So Chris, please share after this. I think it's gonna on the chat room after this, so no problem. Okay, hello everyone. My name is Kai Byun, a global business development manager at Rokit Healthcare. I'm in charge of America, Japan, and Israel. On behalf of Rokit Healthcare, I would welcome you all and thank you for taking your time to attend this second Rokit Play Partners Day. At the start, I moderate the session and do the introduction briefly. Then Chantil will deliver the presentation, followed by Q&A and feedback from all of us. Please note that this meant to generate a free exchange of ideas to build up a good presentation model for bioprinting, which would be helpful to all of us. We welcome an active participation. The session is being recorded for the internal sharing purpose within our Rokit partners. Now, let me introduce our today's speaker, Chantil. Dr. Chantil is the founder and CEO of ONS Bio, distribution partner of Rokit Healthcare, INVIVO Bioprinters, and Humantain Bioinks. He obtained his PhD degree in biology from University of Hanover in Germany and did an extensive postdoctoral research training at the National Institutes of Health, NIH. Now, with the topic names bioprinting and tissue regeneration, recent trends and applications, Chantil will deliver the presentation. Please welcome Chantil. Chantil?
Thank you, Kyle. Thank you very much for the kind introduction. First, let me make sure you're seeing the right version of the presentation. I did the screen share. Can you see and confirm if you're seeing the note side or the actual presentation? Kyle?
I see note side.
You see note side. Oh, yeah. Okay. How about now?
Yeah, now it's perfect. Yeah.
Okay, good. All right, awesome. Okay, all right. I hope everyone can hear. If not, please let us know so we can try to fix that. So, hello and welcome to Rokit Partners Digital Lovers Day. Hello to everyone and good morning or good evening or good afternoon, as the case may be. So today's bioprinting application seminar is aimed at sharing a sample of the latest trends and innovations in bioprinting that has been made possible by our INVIVO, aka Dr. INVIVO 4D bioprinter that we all love so much. And since Kyle has already introduced me extensively, I will skip that in the interest of time. But I do have to thank our wonderful and energetic Rokit team comprising of Yoon Soo Lee, the CTO at Rokit, and our own JS Lee, the Director of Sales, and our wonderful moderator here today, Kyle Beyond, and our very dear Dae, Steve, and Gia for creating these informative slides and the presentation, and being very generous in sharing that with me and giving me the opportunity to present the same today to you all. So from my personal experience, I can tell that the team at Rokit is a very hardworking, passionate, and energetic, and very supportive to distributors and customers alike. So in my opinion, they are the best, and I find it a real pleasure working with them. So without further delays, I will get started with the presentation. Thank you all for joining. So one of the first questions that we all as distributors have to tackle is especially in a customer-facing role, is what is why bioprinting is important, or why even care about it, or why should customers care about it, right? So as one of the key stakeholders in this bioprinting marketplace, that's the question that the researchers and hospitals ask us, and we have to be able to answer. So to answer that question, it's good for us to have a look at the data that's displayed here, which is basically a 2020 current up-to-date data of the top 5 funded disease areas published by the US National Institutes of Health. And clearly, the top 5 diseases are cancer, heart disease, diabetes, obesity, and liver disease. No surprise there. Now, all of these are chronic diseases, and often we associate them with aging. So they are kind of diseases of the aging process. So in 2020, we will also be reaching a landmark where we will have more people that are going to be 65 years than more than the number of people that will be actually younger than 5. So it's going to be an aging population that we will see, a predominantly aging population. And we all can agree that the research addressing the problems of aging and the application of regenerative medicine to tackle problems, it remains as one of the hottest topic of today's science and healthcare. So the first question that we have to tackle, as I mentioned, is why people should care about the bioprinting, right? So again, you also see that the top 5 diseases are also the most well-funded in terms of research funding. So it is important to note that, and also give an impression to the customer that these are areas in which they should be focusing on if they have to significantly increase their chances of being funded for their research. So in that context, bioprinting is definitely going to be a very important technology that they should consider adopting. All right. So one of the major challenges in transplantation medicine. Now, this is to address the question why bioprinting is important, or in what context it is important for researchers to focus. So the major challenge in transplantation medicine is that we don't have enough number of tissues and organs to replace the defective ones in patients. So the 3D bioprinting is pitched as the one promising platform technology that can create these tissues and organs with applications in all these different types of diseases that are listed. These diseases are generally the ones that come with aging. So there has already been a lot of evidence about the bioprinting being an ideal choice for application in being able to print tissue specific manner cells and biomaterials for regenerative medical purposes. So from a research perspective, again, it's conceivable that in future what we will see is that most journal publications will start requiring data from 3D human tissues alongside the 2D in vitro cultures and live animal or human studies as well. So that should make most researchers consider adoption of this technology sooner than later. Now we have talked about the importance of bioprinting here. Now, 1 second. I have a little bit of a glitch here. Bear with me. I will start this again. Am I being audible, Kyle?
Yes.
Okay.
Yes.
All right.
Yeah.
Okay.
But you have to switch the
Switch the okay. Again. All right. Here we go. Okay.
All right. Perfect.
All right. Okay. We are good there. 1 second. Why am I not seeing this here? Bear with me. I am not able to see this here. Okay. My notes page is not moving along as I intended it to be. So 1 second, let me double check.
In the meantime, if you have any question, you can just text here. Text here so that in the end we can do it, the Q&A session. And also, this is being recorded by Chris from Indicate. So the recorded video will be shared in this chat room, so you can download it. And please use it in the internal purposes only for your sales strategy kind of thing. And yeah, Shanti, are you ready?
Yes, I'm ready. I'm ready. I apologize for that little bit of a glitch.
Please go ahead.
So as we have been talking, bioprinting is a wonderful platform technology that serves as a good biomanufacturing technology and has the potential to have applications for all major funded research areas and focusing on various tissues and different diseases. So this is kind of the triad of a combination of cells and different cell types like the ones listed here, plus the ever-growing body of bio-ink innovations that kind of keep these cells happy and nourish them in the printing process and subsequently. In combination with a powerful tool like the INVIVO, which serves as a good platform, we will be able to accomplish manufactured human tissues that can be applied in the clinic for healthcare. So just a brief history of bioprinting. So what we know is that, I mean, the general impression is bioprinting is a very new hot field. The reality is it is already 2 decades old. It began in 2000 with Thomas Boland, a bioengineer at the University of Texas, who pioneered the technology using HP inkjet printer to print bioink. And this was made of living bovine cells suspended in cell culture medium. So since those early days, 3D bioprinting research has evolved and evolved rapidly, so to speak, to include more methodologies from inkjet-based to laser-assisted stereolithography on in-situ printing, and so on and so forth. Now, among them, the extrusion-based bioprinting, which kind of gives a smooth flow of bioink and helps us print layer by layer, is considered as a very useful and popular type of printing. As some of you would already know, that nearly 7 years after the US company Organovo first commercialized 3D bioprinted liver tissues, in 2016, Rokit Healthcare launched the world's first desktop-sized clean chamber included bioprinter, which is our very own INVIVO. So currently, INVIVO is being used by more than 250 labs around the world, and keeping up with this, with its appetite for launching first in market technology, this year Rokit has introduced the world's first bioprinter that's equipped with a carbon dioxide incubator and a hydrogen peroxide plasma sterilizer, which represents a totally new paradigm in the bioprinting space. I know Rokit Healthcare believes that the essence of bioprinter should not be to be just a printing machine, but a true cell culture chamber that can also print. And I think that paradigm is more meaningful and relevant for the clinic. So just to, because Jacob has already given a wonderful presentation last week about the specs and some of the pros and cons of INVIVO in terms of the instruments capabilities and all that. I won't go into the hardware or the software components because it might be too redundant. So the focus on of today's talk is going to be on the application aspect. So before we hit that, just a brief discussion about the advantages of bioprinting that help define the actual essence of bioprinting. First, automation and repeatability and consistency. These are the number one advantages and key features of bioprinting. So bioprinting helps researchers shift from a manual and inconsistent seeding of cells to even repeatable seeding of cells in the proper position. So as you can see from the data here, the systematic seeding of cells using a bioprinter, as shown here, it reduces cell clustering and it also increases cellular viability. And in that process, it produces data that kind of closely reflects the situation that you find in a 3D arrangement of cells in vivo. So the advantage here is, or the bottom line is, that you get very high quality superior data using 3D bioprinting approach for cell culture than using the traditional 2D cell culture. And it is important to also recognize that the cells have higher viability, and it's possible to make structures and complicated networks that would mimic what you find in the INVIVO system. INVIVO system meaning inside the body, not the INVIVO printer in this case. So that is a clear advantage. And second off, the advantage that comes with bioprinting is the ability to do biomimicry, or be able to recapitulate the shape and structure that you find inside the body. So bioprinting is guided by computer-aided design to create a structure that mimics the three-dimensional environment of human tissues inside the body as close as possible. So this is true not only at the level of just architecture, but also in the cellular components. For example, as we can see here, we've got the cardiomyocytes. Let me turn this on. So we've got the cardiomyocytes in 2D. This is a small video that might drive home that point. With cardiomyocytes, the 2D cell culture may create a beating model, but to create a pumping model like the true heart, it takes more than 2D cell culture. It takes more than 3D cell culture. It takes a powerful technique or tool like bioprinting. So bioprinting is the best way to mimic the native structure of target tissue that one is trying to print or one is trying to use in regenerative medicine. So recapitulating the shape, structure of the tissue with the eventual goal of being able to recapitulate the function is achieved by bioprinting. And our best bet today is bioprinting technology. So in terms of the applications of bioprinting, I'm sure all of the distributors here today will agree nothing convinces researchers like a journal publication about the usefulness of a new technology. So according to PubMed today, there are over 1,600 publications on bioprinting. To me, this number looks somewhat being very conservative. We can look into that, but at the least we have got 1,600 publications, and this is growing daily. The applications have come from areas of skin to bone, and also include applications in the printing of many complex tissues like liver, kidney, lung, heart, a whole bunch of those, and even cancer models. So with these in vitro printed bioprinted tissues, the applications are growing rapidly to revolutionize or replace inefficient bottlenecks that are present in existing approaches, especially in areas of drug toxicity, safety testing, or disease modeling, or even in generation of personalized implants and prosthetics and patient-specific implant, as well as in personalized medicine and regenerative medicine in general. So the applications are many, and oftentimes they are just limited by our imagination and the difficulties in finding the right biomaterials. So this slide here shows a kind of because we are not going to delve deep into the specs of INVIVO. This is just to at least give a feeling of the INVIVO bioprinter in action. So I'm sure this audience is all too familiar, and some of you are even experts in bioprinting by now. So I'll be very brief on this slide. What does it look like if we translate these 2D pictures into 3D motion videos? Let's check that out. So this is okay. So here what we are seeing is an actual printing video of Dr. INVIVO in action, and this clip is a technique of printing cell-laden chitosan bioinks into the media. So unlike the traditional method, which is done on a regular petri dish. Now we've got the next one that shows a nerve conduit channel from fibrin, and this is printed as a result of an indirect bioprinting approach called a negative support mold. And then lastly, we've got something interesting that looks more like the fresh form of printing. Here in this clip, we see printing of collagen directly inside a gelatin bath, solving the difficulties that we often encounter with stacking layers of collagen in any structure. All right, so now let's delve into the applications, which is kind of the real focus of this presentation. These are taken directly from publications by INVIVO users, And our team has done a very good job compiling these in the form of one slide each for different application, quite informative and precise and easy to grasp. So these are so basically, if anybody needs more details, then you can refer to some of the citations here and look at the original paper. So first, this is a study of adipose tissue regeneration. There are two major challenges, as you can see here. One being in the existing methods of adipose tissue regeneration. So one is finding the right biomaterial that mimics the tissue environment. Second is to successfully induce angiogenesis in the damaged area. So a research group at the Seoul National University in Korea has developed a new bio-ink with lab-made PLCL and adipose tissue derived ECM that better mimics the physical and chemical properties of adipose tissue. So in the mouse model, when the bio-ink and the fat stem cells are printed with INVIVO and grafted onto the damaged site, there was very good evidence of angiogenesis, as you can see here in terms of the capillary densities, as well as you see the comparison of vascularization between the PLCL versus the PLCL and the adipose-derived ECM scaffolds post-transplantation. And also you can look here some data on the mechanical property of PCL versus the PLCL versus the adipose tissue. So this is a clear example of how new regeneration therapies are being developed by combining tissue-specific biomaterial with the site-specific bioprinting methodology. So moving on to a little more of a adipose-derived stem cell approach. This is another study with the INVIVO concerning adipose stem cells. This research was conducted at Hanyang University in Korea, and it was published in the journal Biomaterials. The biggest problems with the conventional methods of culturing adipose stem cells includes difficulties with in being able to control the mass production and retaining the stemness of the stem cell. So in this study, the research group used INVIVO to create a novel hydrogel-based manufacturing platform, which they call as HES, or hydrogels with an embossed microstructure, which is all kind of explained here. And something that's capable of culturing a large amount of stem cell spheroids. And these 3D spheroids not only had successful differentiation into chondrocytes, as seen by the different marker expressions here, chondrocytes as well as osteoblasts, and they also demonstrated a significantly different profile for the tissue specific gene expression from the in comparison to the 2D cell culture. So the spheroid production using the HES method that's described here, it kind of opens doors to various downstream applications, potential downstream applications, which could include things like cell therapy or ex vivo reconstruction for fat, cartilage, and bone tissues. So that's kind of a very neat way of using the INVIVO for the adipose in adipose derived stem cell research and eventually for therapy. This is another example that was done in the Wake Forest School of Medicine and published in Nature, as you can see. This is an example of creating artificial skeletal muscles. Now, the challenges are there is a need for an alternative to autologous muscle pedicle flap in case of the extensive muscle defect. And then there is also a very low efficacy of cultured cell injections due to low cell engraftment and low survival. These are the current problems. Now, the solution has been to engineer the 3D skeletal muscle construct using a fibrin bioink, including a muscle progenitor cell from human biopsies, and using gelatin as a sacrificial material, and using PCL pillars to support. So using this, they compared to the non-printed tissue resulting from manually seeded cells, the bioprinted tissues, as you can see here in these panels, compared to the non-treated and the manually seeded cells, the bioprinted tissues, they produced about 5 times more muscle formation at 8 weeks after grafting in a rat model of muscle defect. So for future directions, what this group intends to do, or they're planning, is to introduce neurons and neurotransmitters to create a more accelerated model of skeletal muscle regeneration. So again, this is a very nice demonstration of the application of bioprinting to accomplish these goals. Okay, so now moving on to a different tissue type, which is cartilage. As we all know, cartilage is one of the most difficult tissues because once damaged, the cartilage is challenging to repair, and currently it's almost impossible to regenerate. So 2 key factors for an effective therapeutic agent for cartilage regeneration would be to induce the cells to proliferate and kind of home. At the Taiwanese university, Kaohsiung Medical University, the group that did this work, they sought out to develop a bioink for chondrocytes with the appropriate growth factors as well as mechanical strength and a right decomposition rate for the biomaterial that they incorporated. So they actually introduced a photoreactive ink incorporating hyaluronic acid and biotin and streptavidin. So this double cross-linking gel, it offered a very structurally stable and cell-friendly environment for the cartilage repair. And here you will see some of the existing materials or common materials used in printing of cartilage. They come with a set of pros and cons, but none of these are perfect. But this study kind of helps in many ways to find that best approach to solve some of the existing problems. So especially the double cross-linking strategy using the biotin and streptavidin here has helped to add mechanical strength at the same time without compromising the biocompatibility of the material used. So this is with respect to using INVIVO in the context of cartilage regeneration to address some of the existing problems for evolving better scaffold materials with better structural integrity and optimal cell adhesion and viability. This is another example of self-healing hydrogel. At the I mean, the most important thing is the biomaterials are, as we saw in some of the one of the previous slide, among the triad where we have the cells and the biomaterials and the printer together. Developments in each of these is important to accomplish more complex outputs. So in that respect, biomaterials are a very important driver of innovation and applications in the bioprinting and tissue engineering field. So currently, in spite of the rapid development of 3D printing in terms of the instrumentation and cross-linking techniques, there is still a struggle and an ongoing search to find a bioink that requires no additional manipulation or cross-linking induced by UV or temperature or chemical, because these are somewhat undesirable because they can damage the cells. So the ideal method would be something where the biomaterial can cross-link without the use of any of these agents. So with that in mind, the group at Hanyang University in Korea, they developed a self-healing hydrogel called the OHA, and that the OHA stands for the oxidized hyaluronate and glycol chitosan, and in the presence of adipic acid dihydrazide. The names are not important, but the point is the hydrogel that they came up with forms bonds instantaneously without post-gelation or additional cross-linking processes. And that's kind of demonstrated here. They cut these and then they let them heal, and within less than 10 minutes, they're able to heal. And this is a very close microscopic view of that process of healing of the hydrogel. So as you can see in this picture, when they put these chondrocytes into this gel and encapsulated them and cultured these gels for a week, they expressed the typical differentiation markers like SOX9 in this case, in the graph here, and at week 3 they expressed the COL2, which is the genetic marker for cartilage differentiation. So that demonstrates the usefulness of this hydrogel in spite of the innovative self-healing property that it has. So it doesn't compromise the ability to serve as a good differentiation scaffold. So going forward, this self-healing hydrogel system is likely to have a potential in various other tissue engineering applications, possibly in drug delivery systems. This is another example of self-gelling material that can do so without the use of any photo initiator. Silk, as we know, which is kind of used in this study, which was done in DGMIF, the Daegu-based Medical Innovation Foundation in Korea. Silk protein is made of fibrin and sericin, and it has always been viewed as a promising bioink candidate because it's known for its anti-UV bacterial and inflammatory properties, and it has a very good effect on cell regeneration. So this group combined this new bioink, the sericin protein from the silk, with gelatin and glycerol, and came up with this self-gelling biomaterial. And they were able to confirm that the gel can create high precision scaffolds, as you can see here, which with very high resolution of 200 to 500 micron pore structures. And they also assessed the cytotoxicities as seen in this panel. So all of that checks good and promising. So beyond the bioink applications, the sericin is also being investigated as a wound healing ointment and a patch material owing to its intrinsic healing properties. So that could be an active area of pursuit for this self-gelling biomaterial in the coming years. So this is yet another example of innovation using the novel materials and using INVIVO to print those. This is about a very interesting case as well, which is the wood-based biomaterial. I mean, despite the many advantages we talked about when we talked about silk in the previous slide, it does come with limitations of being very expensive and very rare to process for bioink usage in a large scale. So many researchers are now looking to materials that are more abundant and natural, like plant and wood, of all things. Plant and wood derived materials are xeno-free, so that's good for regenerative medical applications. They have a reduced risk of immune complications or immune rejections compared to any conventional chemically synthesized or animal derived material, and therefore they are explored as a clinically feasible option. An INVIVO user, in this case from Finland, Abo Academy, they developed a wood-based bioink containing cellulose nanofibrils and galactoglucomannan methacrylates, GGMMA. So it's a nice long acronym. So the in short, what they have confirmed is the printability of the bioink by printing various scaffolds. And they also have further tested its ability to support various cell types from skin to pancreatic cancer cells by comparing the cell proliferation on the seeding matrices printed with various inks and looking at different times of incubation. So this way, this special biomaterial that they have created, CNF GGMA ink, it broadens the map of bioinks, which potentially meets the requirements for a variety of in vitro cell matrix and cell-to-cell interaction studies, particularly in the context of tissue engineering, cancer research, and also possibly high-throughput drug screening. Here we've got yet another. I mean, each one of these studies is really exciting to talk about because the applications are so varied, and the problems that or the challenges that they address are different, but all with the same INVIVO bioprinter. So again, it stresses on the point that we talked about earlier, INVIVO being a good, powerful platform technology for various applications in various tissue types. So briefly, in this case, it highlights the fact that INVIVO can be used in surface engineering and coating technologies, not just printing scaffolds and structures. So in this case, it's been done to improve the cell-to-cell interactions. So a group at the Seoul National University, they have developed a novel hyaluronic acid catechol coating platform, as it is called. It's the acronym is Nature-Inspired Catechol Conjugated Hyaluronic Acid Environment, NICE. And it helps to boost the salivary gland tissue engineering efficacy of the previously reported materials by using this as a coating. So what they have compared here is conventional PC membranes, which I believe in this case is shown here in the figures here. PC membranes agarose as a, so the group basically compared the conventional PC membrane agarose as a key member of the hydrogels, and they 3D printed PCL scaffolds as a representative of hydrophobic polymers. So the result showed that regardless of which surface regardless of the surface, there was a very high amount of salivary tissue budding happening compared to the bare uncoated. So this also kind of shows that there is enhanced vascular endothelial cell growth, as you can see in the branching here, using the niche coating. So the group is now, and this is kind of a quantification of the budding levels to quantify the amount of budding in the coated versions. So the group expects that the coating will expand the range of biomaterial candidates for researchers doing salivary gland and other epithelial organ tissue engineering. Here we've got the cancer studies, an interesting cancer study. So the basic story here is that we all know that the 2D cell culture is not a good representation of what happens when cells are in a 3D inside the body. And that is applicable also to the tumor microenvironment, not just regular tissues and organs. So when you are looking to develop cancer therapy, it's very important to understand the tumor microenvironment as close to reality as possible. And that is not possible using 2D cell culture systems, because they can provide very unpredictable and misleading data about in vivo responses. So therefore, we need a 3D cell culture environment that is necessary to mimic the tumor characteristics. So in this study, what has been done, which was conducted by Jun-ong University of Korea and Wake Forest as a collaboration, Wake Forest Regenerative Medicine Center in North Carolina. INVIVO was used to develop a 3D human bladder cancer model, and it was compared with the 2D model, as you can see here, to evaluate the behavior of two different cell types, the 5637 and the T24 cell lines. And what was, as one would expect, the results of the chemotherapy tests using, in this case, rapamycin and its effect on the mTOR pathway. It turns out that the results of the chemotherapy showed different profiles, as you can see in this Western here. So different profiles of the reactions regardless of the cancer cell lines. Therefore, the group concludes that using 3D model for cancer is crucial to better assess the drug's efficacy, and it's time to seriously think about doing high throughput screening on 2D cell culture models just for the convenience and the fact that we are so used to doing that. So we can expect to see more and more labs in this area kind of transition into a 3D bioprinted approach to understand the cancer drug screening much better. All right, so this one is about not just application of, so far we talked a lot about the application of in vivo in cell scaffolds and biomaterial development. Now here is a very interesting situation where you can also use, or in this case it's been used, in vivo has been used for bioprinting a thermoplastic elastomer, that's shown here as TPE. So in this study that was conducted at CRICT, it demonstrates the creation of an ergonomic bracelet, as you can see here in these images, using a petrochemical thermoplastic elastomer and something that's combined with the biomaterial called nanocellulose crystals in smaller quantities that can kind of homogeneously distribute the nanofiller, and it affects dramatically the strength of the thermally 3D printable biofuron-based TPE, thermoplastic elastomer. So compared to the TPE alone, the bio-based TPE demonstrated greater tensile strength and adequate heat response, as seen here, when voltage was applied. Now, this research is a very good example, an illustration of using the 3D printing technology to innovate medical device and wearables development. So, which is another hot area we all know. So, what we see in this example is INVIVO kind of crossing into the other material science areas in its potential to impact. And this technique can also be used potentially to create a cancer cell-like environment for a drug screening platform. And those are kind of the future directions this group is looking into. Oh, this is also at another material science related approach, but in this case it is little bit of a deviation from the biology aspect that we've been focusing heavily on so far. In short, INVIVO has found applications in customized filtration devices with applications in radiation safety and radiation hazard mitigation. Usually ceramic absorbents are used for absorbing the radionuclides in managing nuclear waste. They are very difficult, brittle, and expensive. So what the solution that has been tested in the study is, and this was done at KAIST, is the development of a ceramic slurry ink that is zeolite based, using a 3D printing process to create filters such as these that can be made on demand. And their conclusion has been that based on the study, that the 3D printing can offer a method for manufacturing adsorbent filters using the zeolite material that they've used here, with the good control over the architecture and the porosity, and also minimal costs overall. So the 3D printing of materials can be used in development of many novel filters such as these, with varied applications where one can control the porosity at will. So such ideas are truly what to say, there are so many things that can be done, and as I mentioned before, the applications are limited oftentimes by the imagination and what materials we have at hand. And it's only conceivable that in the coming years we will see more and more novel applications that we wouldn't be able to imagine today. Okay, this is a INVIVO application as well, where this is about the synthetic bone graft printing. So the Korean FDA has approved a 3D printed bone regenerative scaffold and implant printed using the INVIVO bioprinter sitting here. First challenge is that the difficulty to find a low cost and a customizable, patient customizable bone graft. I mean, this one size fits all approach that we've been relying on is expensive and not perfect. So that is the challenge that we are trying to tackle here. So in this case, a combination of PLLA and HA were combined and printed with Dr. INVIVO's hot melting pneumatic dispenser. And I think this is a very good point to talk to some of the customers working in this area. And the result was a very good, neat, customized synthetic bone graft that could be applied to various indications as bone substitutes. So these bone grafts would be applied to not only orthopedics, but as we all know, yet another low-hanging fruit application of bioprinting, dentistry. And these are some of the data in this work that show the ultrastructural patterns in morphology as analyzed by X-ray image, and they are quite impressive. So this is again a good example of bioprinting platform and its commercialization potential. So to address the challenges of this existing one-size-fits-all cookie-cutter bone graft approach that we currently have, this company BioOrigin, they have made a leap to customizing using 3D bioprinting, and this kind of offers a new hope in tissue repair, especially in the craniofacial and dental bone loss from oral cancer or aggressive oral cancer that more often than not require customized shape repair. Okay, now we are getting to the almost to the end of the presentation, and this is our Rokit INVIVO in action, and these are projects from our own Rokit R&D, and the current big success, feather in the cap of Rokit, is the successful clinical trial for diabetic foot ulcer. So in this slide and the next two slides, I'll be introducing the works by Rokit Healthcare's own R&D with INVIVO and advancing the bioprinting to in the area of novel therapy development. Because what's more important is not just being able to do things for research, but actually moving some of that research and applications into the clinic for patient care. So diabetic foot ulcers, they represent one of the biggest debilitating disease in the world. And India is a country that has many patients with this condition. And in the last year, Rokit Healthcare did the first clinical study using Bioprinter INVIVO to regenerate the skin in diabetic foot ulcers. And the results are kind of presented here. Long story short, what we see here is the leveraging of a patient's autologous cells with the bioprinting technology and developing a patient-specific, site-specific treatment with no immune rejection. So the results were so encouraging that it took, on an average, only 4 weeks to close the wounds, which were until then open and healed for months to even years. So that is a tremendous achievement, and we can envision that this therapy will be applied to more skin conditions, from burns going all the way up to scars caused by bed sores in hospitalized patients. So this is a truly good example of INVIVO not just in research, but also in the clinical practice. And this is yet another work that's currently being pursued in collaboration with Mass General Hospital. This is in the context of cartilage regeneration. So the goal is to use autologous rapid bioprinting approach to generate a customized solution for patients. One of the big problem is the degenerative arthritis is becoming a widely occurring disease that's very closely associated with aging. And the hyaline cartilage is the key to treating the arthritis, but the self-renewal of this is almost impossible. So conventional stem cell treatments have a relatively low efficiency and efficacy compared to the amount of loaded cells and the ambiguity about their stability and distribution. So Rokit Healthcare, what they have done now in collaboration with MGH is to conduct a preclinical testing of the cartilage regeneration using the Beagle models shown here, and using the stem cell laden bioprinted matrices that are printed. In these cartilage defect models, the results have been very encouraging so far, and quantitatively they've been able to demonstrate that hyaline cartilage regeneration is localized in the knee, and qualitatively they observe that the beagles are recovering and having restored walking functions. So that's with the clinical application of INVIVO work done by the Rokit R&D, in-house R&D, with respect to cartilage regeneration. And this is moving on to the last application that I'll be discussing today. My personal favorite, which is the hard patch regeneration. Again, this was done in collaboration with Fraunhofer in Germany. One of the big challenge here is that the ischemic heart disease, they are caused by when a blood vessel fails to kind of constricts or is lost due to death of tissues, and that results in lack of supply of nutrients and oxygen to the heart, and that part becomes necrotic. Now, what we have kind of known from medical practice is that if we replace the necrotic part with the heart patch, which can synchronize with the rest of the organ, then some of these ischemic symptoms at the necrotic site can be resolved and proper heart function restored. So in this study, INVIVO has been used to develop a patterned disc that allows the induced pluripotent stem cell derived cardiomyocytes to align and create a beating model, such as this one here. And this is a real cool video. I think this is kind of the money slide, I would say, except that it is the very last slide. So this in vitro heart patch has been made using INVIVO, and it's interesting to note that this has been beating for at least 6 months in an in vitro culture, demonstrating how robust the preparation has been, and how friendly the printing process has been to keep the cells viable that long. So to summarize, there is going to be a lot of development of biomaterials suitable for this type of application, things that are biocompatible with very high potential for treatment of chronic ischemic heart disease. And to summarize the presentation, in the very beginning, I mentioned about the bioprinter being a platform technology. Now, when it comes to choices of cells, bioinks, applications, the possibilities are limitless. A combination of innovative thinking and materials and the printer and researchers, you can imagine that there are many problems that can be tackled. But having said that, that optimism should also be balanced by some of the continuing severe challenges that we have to overcome until the bioprinting tissues become reality and something of a routine in the patient care. So the good news about all of this is that so far we have seen so much progress happening within our own INVIVO community of users and the Rokit's in-house R&D. Now, all of this is possible because of the passion that each of these groups and Rokit has, and their commitment to making bioprinting application in the real world a reality. And I think as distributors, we distributors and partners of Rokit in this incredible journey, if I may call that, we can provide guidance and motivation to our customers by sharing some of these knowledge that we have accumulated or come to know of, to help them make life-saving and life-impacting discoveries that will use bioprinting and kind of shape the way in which medical practice is done in future. So that's kind of my conclusion on this. I thank you all for your patience and joining me today. I hope I made some sense. And again, I thank Kyle, Dai, and the rest of the team for putting together a wonderful presentation and generously sharing that with me. And I intend to use this for an upcoming customer presentation as well. So this kind of was a helpful rehearsal for that, because I've never handled these new set of slides. Thank you. I'll be happy to take some questions.
Yeah, thank you for the great presentation with the new slide. And as I mentioned in the chat room, if you need the publications papers, it is on our website. And other than this slide, the PPT slide papers, there is also other publications too, so you can check that out too. So if you have any questions, you can turn on the mic and then come here, and then or so you can text here. Dae, do you have any feedback or questions from there?
Hey, I think overall it was good. I like that you were basically able to describe each one of the applications from from the point of view of a researcher, and how to explain the data, comparing the challenges of the existing technologies with the benefits of bioprinting. So I think it would be important to really stress that all throughout. Like, why should a scientist abandon his or her own existing methods and adopt the 3D bioprinting technology? And I think that should be stressed throughout all of the applications. My other feedback is, I think in general, it's helpful to have a presentation within the limits of 30 minutes and leave the rest for Q&As. One that leaves a lot of, well, you know, a little bit of mystery so that people can address questions at the end. And,
Agreed.
Yeah, and second, that the keeping it within 30 minutes, I think, could keep the audience a little bit more engaged.
Okay.
So I think as long as we work on that, I think it'll be a great presentation, and you'll do well as always in front of the audience.
Thank you. Yeah, this I realized it was getting longer, but there was so much content in there, and you guys have so many applications. I think it'll only get more and more difficult with more and more publications.
But
Yeah,
I think based on the customers, his or her specific interest, maybe we can digest the slide, and then also like in the first part before the applications, you can a little bit, yeah.
Okay.
Sure. Those parts, yeah. And the PPT slide was good because it includes applications not only biology but also
Material science, so on.
related polymer. But you know, sometimes you know it's just for the just interest sometimes because they need specific information other than that. So maybe you can just introduce, but you don't have to, you know, explain everything. So yeah, maybe you can digest it like that. Yeah.
Agree. This was the kind of envisioned for the distributors in mind. So they would, if people haven't seen these slides before, they may have a broader overview of different applications. But agree, depending on the audience, we may have to take out some slides and focus on only what's important for them.
Yeah. Yeah, I think this presentation was really helpful because we can it's references of our sales, actual sales, and then we can apply this presentation to when we do the presentation to the customers. So thank you. I really appreciate, and we are really appreciate too. So I think because of the delay, I think we have to, you know, wrap it up. Oh, hi.
We have somebody.
Hi.
Hello.
Hello, Kyle, Daya, and
Hello.
What's your name? Dr. Is it
Senthil.
Kelvin? Senthil? All
Yes,
right.
Senthil.
Yeah, I know you guys are wrapping up. I think I'm kind of late in jumping in. I actually like went through like half of the slides toward the end. It's really exciting for me because I'm kind of new here. So can I ask a few questions before you guys go? Would that be okay?
That'd be fine. Go for it.
Yeah. So yeah. First of all, can I actually have recorded video of this after this? Would that be fine as a distributor to have it?
Yeah, it's will be on the chat room, so you can download it.
All right, great. And secondly, I have a little confusion when people are like, for us, when we print it out, we need to use the scaffold, right, to get the cells viability, proliferation better and all. So when we actually come up with that scaffold, is that like outside of a human body application only, or it can be attached within our body that it will dissolve all the scaffold, like the silk protein, like a lot of scaffold, right? Can you explain a little bit on that?
So if you look at the bone graft, the synthetic bone graft slide, there is a polymer called PLLA.
Yeah,
Shelley, can you show it?
PLLA and HA. Yeah, I remember that slide. The PLLA and HA, right? Yeah.
So that would be the example of making scaffold outside and then go into the body. Yeah.
So some of the scaffold are like two categories, right? One is just for in vitro kind of like testing. One is for clinical approved.
Yeah.
So it depends on the material.
Yeah, for the in vitro toxicity test, they just use it in vitro. But
Okay. So in other words, all the scaffold that are being printed out using INVIVO, they are definitely non-toxic to cells, but not necessarily it can be non-rejected from human body. Is that
No.
something? All right. Okay, and then quickly to go to, so when we talk about bio ink, I'm not really sure that it's like, when we print it out, it's limitless by our imagination and material that we have in hand. I noticed that. But then so far, the bio ink is actually being developed by the researcher themselves, or they can actually purchase from Rokit, some of them, or
So-
what the category of the bio ink?
We have INVIVO gel, which is gelatin based. That's the for now. That's the only one available bio ink. But we are obtaining the other product line from KIST, and I will let you know the timeline after this session.
Okay. All right.
I will have to check and get back to you.
Yeah, and then I just a little bit more comment on Senthil's presentation. For me, I think it's really helpful for me just now, I think for the distributor, because we're like lack of knowledge on what kind of the various application that people are doing out there. And like, for example, yesterday I had a meeting like for like almost 2 hours with this doctor that interested in our INVIVO. So I showed her all the applications, some of it like already have in the slides. And so that's really helpful. At least I know like that's a slide, and what's the background of it, I can't really sure. But then they just have a look at it, something like that. Now I get an idea. But it will be more helpful if I could know in the feature of our INVIVO, we have the syringe, the filament extruder, and pneumatic dispenser. I really love the way you present the synthetic bone graft, right, for the commercialization, and they really need the pneumatic, this hot dispenser in this case. So then I can see the feature that we have in our INVIVO perfectly suit for this kind of application. Like just now you went through the application a lot of that, like this one they do this one, this, but I don't know how when they're using INVIVO printed at which aspect. Like I know that you printed out the graph, but then do you use the filament? So I'm like in the question mark, like how do you do that again? Like I know you can print out, but what features? So then when I talk to them, look, you need this feature. You cannot just buy upgrade, for example. You need the pneumatic. So I know which one that I can learn throughout the timeline. When I keep on like engaging with you on the presentation, then I can integrate INVIVO features with the application. Yeah, that would be helpful for me.
That's a perfect suggestion. Yes, I totally agree. I'll try to incorporate that for my future presentations. You're right.
Thanks so much.
It's important to point out which of the print head has been used. Good.
Yeah, so it's a little bit, just maybe just up like 5 seconds, 10 seconds. Oh, by the way, this one, it uses the syringe with a, like -5 degree, for example, for collagen. Like at least I have an idea. Oh, so doctor, you need to use this. So like, yeah.
Right, I agree.
That's all.
I totally agree.
Okay.
Thank you for the feedback.
Yeah, sure.
Thank you so much. The assumption was wherever it's soft hydrogels, then it's kind of obvious that they would use syringe dispenser, but it doesn't have to be inferred. Instead, I could make it more loud and clear. So thank you for that suggestion.
Thanks so much. Yeah, I would really appreciate. Yeah, thank you.
Sure.
That's all from me. Thanks very much.
Thank you.
And where are you from?
Oh, I'm from Kuala Lumpur, Malaysia.
Oh, you're from Malaysia. Okay, good.
Yeah.
Thanks.
Thanks so much.
Thank you so much.
All right.
We'll have that feedback and edit a little bit and show how it works with the INVIVO in the slide, and then maybe we can share it to all of us. Yeah.
Yeah, perfect.
Right. Yeah. Okay, that's all from me too. So Shanti, any last comments?
Well, thank you so much. If you have any other applications that you know of, please share with us. And any other suggestions that you have not been able to express today for whatever reason, please send a text or email.
Okay. All right. Thank you, all of you, and I hope you enjoy the rest of the day or have a good night. It's a global conference. Thank you for your time. And
Thank you all. Thank you all. I really appreciate, and thanks to the team for putting these slides together. So it was a good exercise for me, I think.
Thank you. Thank you, guys.
Thank you. Bye.
Thank you.
Have a great day.